Let's start with some science. I know that it's maybe a little bit more science. I don't know. I try to be as simple as possible. So I'm talking a little bit about how we can target metabolism in triple negative breast cancer. We know that every cancer, every cell requires metabolism or energy for their survival, because we need energy. Cancer cell need energy. Any cell need energy. So one of the targets, or one of the emerging targets in cancer field, is the metabolism. How we can target the metabolism. This cartoon actually, I think I show every year, that talk about mitochondria, which is one of the a major part of the cell which produce most of the energy. It's all the powerhouse of the cell. So this cartoon is saying is we get mitochondria from our mother because it's a maternally inherited gene. So that is why most of the time we say it's many of the maternal diseases we use mitochondria as a sequence as a tool, because it's mostly nowadays we say that there is a paternal DNA inheritance, but mostly it is considered as a maternally inherited gene. Okay. So I'm talking more about triple negative breast cancer here. And in this group, I think there is no need to explain what is triple negative breast cancer, which is mostly hormone receptors negative and HER2 negative tumors. That's why they are called the triple negative breast cancer, or it's also called basal subtype, because basal subtype is having a lot of character which is very similar to triple negative breast cancer. Why we are giving more importance to triple negative breast cancer is for ER positive and HER2, there are at least some specific targets available, but triple negative breast cancer is currently really suffering from lack of any targeted therapies. So they are mostly treated with the chemotherapy, regular chemotherapy, surgery, and nowadays there are few drugs approved, but they are not. There's a lot of opportunity to treat triple negative breast cancer is missing now. So we need to do a lot of research in that area too. And also one of the reasons is triple negative breast cancers are usually aggressive, and they are also more prevalent in the African American breast cancer patients. So there's a little bit of racial disparity in triple negative breast cancer, especially in their aggressiveness and treatment response. And I was talking about mitochondria. So this is if you think this is a cell, the mitochondria is sitting something like this out of the nucleus, and that makes it's like the powerhouse of the cell. It makes a lot of the energy. 90% of the cell's energy is produced in that mitochondria. And the interesting thing is, in the cell we always have the DNA in the nucleus, but mitochondria is the only organelle other than the nucleus that has the DNA. Mitochondria is like a bacteria, like they have their own things, they function by themselves quite a bit, semi-autonomous organelle. So that's why it's a very interesting organelle, because it just the mitochondrion has its own DNA. So this slide, it's complicated. I just wanted to tell that if you consider this is a mitochondria, from a long time people were using all our energy is coming from glucose, but now we know that it's not always true. The mitochondria can make energy also from fat and also from glutamate. So our previous concept was all the energy is through glucose, but now that's not true. Especially in triple negative breast cancer, they use a lot of fat for the metabolism, for the energy. So our lab, we always do mitochondria, what's the function of mitochondria? For example, this is a classical figure. We change the mitochondria in the same cell. We put different one from a normal, the green color is a normal or benign one, and the red color or the reddish color is in a cancer. So if you look at this, they have the same nucleus, but we change to a normal mitochondria, and we keep the cancer mitochondria here. And if you look at the mouse, when they form the tumor, you can see that every mouse gets only one-sided tumor. Even though we put both in the same mouse, none of these mouse are forming tumor on the other side. That's where we have a normal mitochondria. This suggests that mitochondria is very, very important factor in the regulating the cancer formation. So that is why we do a lot of research in this area, because mitochondria is a hub for metabolism and also a major regulator of cancer. So today I talk a little bit more about metformin. I think most of you know metformin, which is an anti-diabetic drug, and it's a well-known drug. Most of the type 2 diabetic patients, they take the metformin. But there are some recent studies, they suggest that metformin also can be an anti-cancer drug. The reason we thought that in some of the larger studies, in the patient studies, they found that those who take metformin has a lesser incidence of breast cancer. And also, there is a reason that because metformin is controlling the glucose, and which is a major source of energy. So there are a lot of studies, a lot of clinical trials, for example. A lot of clinical trials are going on metformin, and some are completed, some didn't give a major impact. But even still, in many cancer models, metformin is in a clinical trials are going on in patients, because it's a very easily available drug, very economical, and also its a safety factor is well studied, because metformin in the market and with the patients for long time. So there is no major safety issues with metformin. The problem is metformin is alone. Is it alone enough to control the cancer? That's the problem, because many of the clinical trials didn't give a good outcome. So my lab was mostly focused what will be a combination we can use to metformin because metformin, if that can be a supporting drug, it's easy available and it's very economical, and and there is no need to wait for a long time for clinical trials to come to the bring it to the clinic. So we were more focusing how we can make it more a useful drug in breast cancer. That's what. So what we did say metformin single trial drug trials in our this is a patient derived xenograft. That means we take the tumors directly from the patients and we grow them in the mouse, and we never culture them outside. It's always mouse to mouse. These tumors maintain quite a bit of similar characteristic of a female tumor, or sort of the breast cancer patient. So we treated them with the metformin, and we see sometimes there is a reduction in their tumor growth, but it's very, very, very minimal, suggesting that in some of the cases there is no effect for metformin. Some cases there are effects. So that suggests that metformin alone is not doing much in this type of cancers. These are mostly triple negative breast cancers. So we were looking what may be the best option, and the reason is there were some studies which they conducted, and the major papers saying that there's concentration of metformin is an important factor, because depending on what concentration we take the metformin, there is a big difference in their action. In our lab also we looked into this factor, and we found that. This graph, I need to explain that this is how the cell is viable after the treatment. If you understand, this is the concentration of the metformin, and this is the viability of the cell. So if you can see that in the beginning stage, that means these are the lower doses. At the lower doses, actually the cell's viability is increasing, but in the higher dose, it is coming down. This is the same thing. The lower doses, it is increasing. So that means in a lower dose, it's doing actually it is doing opposite. It's again same here. So this is fenformin. This is similar like metformin, little bit more potent drug. So both fenformin and metformin showing the same pattern. So that means at a lower dose, they support the tumor growth, and at the higher dose, they decrease the tumor growth. So we realized that they have a different mechanism depending on the dose. There is also a clinical trial. What they did is they treated the patients with the metformin, and they took the patients before the treatment and after the treatment, the tumors, and they checked for that. I don't want to go more detail into this science part, but all I wanted to say that many of the proliferation, that means the cell's growth pattern in many tumors, it is going up after the treatment instead of coming down. So exactly like something what we observed in our previous study, the amount of metformin getting into the tumor may be less. That is why they are showing a opposite effect. Because there are some cases they go down. The blue lines you can see they are going down, but the thing is most of the red lines you can see the proliferation is after the tumor. These are the patients after taking the metformin for 21 days, they are getting the opposite result. So since metformin is a likely anti-cancer drug, but it is not giving the desired effect in the patients, we decided to look what may be a better combination with the metformin. So we call this as a clonogenic assay, where we treat the cells with the different drugs, and we see how they are. Each ones are colonies, they are forming from the cells. So you can see that this is a fatty acid inhibitor. I told the triple negative breast cancer take a lot of fat for their metabolism. So the fatty acid inhibition is actually reducing it. Metformin is slightly reducing it, but this combination is really, really working well. So this is different cells. You can see that it's always the fatty acid alone is not, inhibition alone is not working well. The fenformin or the metformin alone is not working well, but their combination is working well. This is a graphical representation. You can see this is the control and this is individual drug, but this is a combination. It's a huge effect we are getting in the combination effect. So we found that may be one of the combinations we can use. And we also know that one of the target of this fatty acid is a SERC, which is a cancer pathway. So we decided to stop that pathway instead of fat. So we did animal experiments. We did a similar experiment using that the dasatinib, which is it's anti-cancer drug, which is available in the clinic. So we combine these two, then we can see a similar effect where, when we combine them with the fenformin or metformin, when we combine dasatinib alone, they are not giving much effect, but the combination therapy is giving a good effect. So then we wanted to look what happened to the mouse if it is the same thing. So we went to the mouse, and again, if you look at this, you can see that this is a tumor growth, and this y-axis is the tumor growth, and this is the days of treatment. You can see that the black line is the vehicle that they are getting placebo. There's no drug, and this is the red line is actually the metformin. Blue line is actually dasatinib, which is a SERC inhibitor, and this green line is the combination. So you can see that the single drugs are growing, quite a bit fast, just like the controls. There's no much difference, but if you give the combination, there's a huge effect. This is, for example, this is a survival, the percentage of the survival. You can see that the single drug, so the no drug, and the single drugs, and the combination has a huge effect. We see this is another PDX model. I told that this is from the patient, directly from the patient, and again the combination is having a huge effect, and the survival is better. Okay, so that suggests that when we combine the metformin alone may not be a good target, but if metformin can be combined with the other drugs, like the SERC inhibitor, that can give a very good choice. So when we think about the availability of the drug and the safety factor of the drug, I think this combination is giving a big advantage for the treatment. But again, we know that the most of the breast cancer patients, they are not getting major problem not from the primary tumor, but from the metastasis. So we actually did exactly similar scenario what happened with the patient. So what we did is we generated the breast cancer in the mouse, and when the primary tumor they developed, we removed, just like we do for the patient, we surgically remove the breast cancer. And we know that after some time, sometimes when we remove the breast cancer, they develop metastasis. So we did exactly similar ways. So after that, the mouse don't have any primary tumor, we remove them. Then we treated them. So then we look for what happened to their lungs or metastasis formation. So we did that. So when we remove the tumor, you see these are the tumor size. So we remove all the group. We remove the tumor at the same size. So they all have similar, almost similar tumor size, because we didn't want to treat early so that one will be small tumor, one will be big tumor. So we remove the tumor at almost the same time, and then we treated. Then we waited for the metastasis formation of that mouse. Interestingly, we can see that this is actually, if you look at this, these are the lungs. So if you look at these lungs, you can see that these vehicle treated or the control mouse of lot of lung metastasis. The metformin alone, they have metastasis much lower, and this is dasatinib alone, they have metastasis. But you see these tumors, which is the lungs, which is from the mouse, which got combination therapy, their lungs are very clear. There's almost no metastasis. If you look at the pathology slides, you can see that there is in the lungs, there are metastatic tumors, and there is metastatic tumors, but the combination therapy, the mouse, the lung is very clear. They don't have any metastasis. So this is a representation. So the zero is almost black color, and the increasing the color means more metastasis. If you look at this one, you can see that the combination, I think most of the mouse don't have any metastasis. They are completely empty. So I just wanted to say that some of repurpose, we say it is a repurposing of the drug, like means whatever drug is available for other diseases, we may be able to repurpose it for the cancer therapy. So we are looking into that direction, because a lot of metabolic inhibitors are treated for other diseases. The advantage is they are already in the market, they are already in the clinical trials, the safety factor is well known. So our question is whether such drugs can be repurposed instead of developing a new drug and waiting for 15 years to get the clinical trial and finally get into the market. So that is one of the strategy we take it in our lab, and we try to target many metabolic inhibitors which are already available, and also we are looking for some of the antihypertensive drugs which is having a metabolic effect, and to see whether they can be repurposed instead of we develop a new compound. With that, I stop here, and this is my lab, and then the people who did the work in the lab, and especially Dr. Park, who did most of the work which I was talking today, and these are our funding agencies. Thank you. And please let me know if you have any questions.
Hi, Dr. Benny. How are you?
This is good. How are you, Lopa?
I'm good. So no, very interesting work, Dr. Benny. So, and it took me back to my days in the intensive research in the lab. So I worked a lot with OT1 and MDC31. So I have a few questions about this. But this is so interesting, and we have so many patients, with TNBC in our support group, and so it's still in the is it still in the bench, or you have taken it to the clinical trials also?
The metformin is already under clinical trial, and many metformin clinical trials fail. And I think I showed you there are many cancer models they are doing metformin as therapy, but this combination noted, we are actually, this is a very new data actually. We are trying to
publish. With dasatinib, right? So this is so interesting that because you didn't see any lesion in the lung, and my question over here, Dr. Benny, is that, so what phase of, if you are thinking of taking it to the clinical trial, are you planning to, give the dose of metformin along with the, is it like any other type of chemo, or specifically to dasatinib, and which stage, like for example, if it is a neoadjuvant stage, or like before surgery, post surgery, or once, that's where my question would be, because we have patients, and they are, if there is anything with TNBC with metformin on the trial that you know of, if you can share with me, because I can, and is it also with the metastasis cases? Because there are relapse also in our support group. There are cases diagnosed late, or it is also applicable to the early stage diagnosis. I'm just trying to translate it to how we can help our patients.
Yeah, that's very interesting question, because the reason is that the whole point of we are doing it that because sometimes in a research field using a old drug is not an attractive thing, like, because the companies are not interested, because their company is not getting much benefit out of it because it's out of patent. And in my case, for example, this metformin is out of patent and dasatinib is out of patent. Like, they may not be able to, they may not be excited in this, type of studies because they are not new drugs. But my question is, as you said, whether I should wait for another 20 years for getting a new drug, or I should use something which is already available. Because we need data as early as possible. Like, we need the result as early as possible, right? So that's our question is. So there are a lot of new drugs are coming. I'm not saying it's not there. Every time new drugs are coming, but the reason I chose metformin like a drug is it's very safe. Everybody takes metformin like a market.
Yeah, it's for the diabetes. Correct.
It's a very routine thing for most of the patients. Like in the people, like if they're a cancer or not. But again, as I mentioned, the problem is the dose is a factor. If you take metformin alone, because it's not how much you take, how much the tumor gets. That's the thing. Like, because the effect is depending on how much it gets into the tumor. Right. So that is why our initial study was to find out a concentration dependent way. Then we saw that if the tumor is getting very low concentration, that it might do an opposite effect. Like, because I think, I hope you remember, I was showing it was like, the beginning stage, it was increasing.
Right.
So, and also, if you give even if you give a reasonable dose, it activates some other pathway to escape from that, escape. So that is what we wanted to give a combination to stop it. And I think these combinations looks very interesting for us. At least the lungs are very clear. Yeah. And also we actually try to do exactly like, we generated the tumor, we removed the tumor, and then we treated just like what happened to the patient. So your question of whether it's a neoadjuvant therapy or adjuvant therapy, we did both. If you look at it, like, we treated the primary tumor, we also treated the metastatic stage. It should be okay, but I think we probably have to do a few more, larger scale studies on this one to find out what it is. But it's very interesting, very interesting.
Yeah, it's very interesting. And when you said about the dose, right, one more question that comes to my mind, Dr. Benny, is that because, for example, did you see a difference? Like I know most of many population with diabetes, they already have, they are on metformin, right? So did you do any study where you are already a patient group who are with breast cancer, TNBC, already on metformin because they are diabetic, versus a patient, because did you see, or do you think in that direction? Because that would be another very interesting catch. That is one thing I wanted to ask. And the other thing is that while going through, I really loved it, what you did. So my other question would be, when you are talking about the dose at high concentration, what is the toxicity level? Because is it like beyond the threshold that our human consumption or during diabetes that we are already taking and that is FDA approved, or you're going way higher so that it's effective in the tumor, and that dose, will that be FDA approved or is already in the approval panel?
Actually, we didn't go very high. In our case, we were trying to match somewhere in the human approved dose range. That is why I'm saying, so we can, the thing is, Lopa, we can probably kill the tumor by dumping that drug, right? But we didn't want to do that. So that is why we were converting the human equivalent dose of the mouse. That's the way we are doing it. Yeah.
That's good. That's good. And please keep me on the loop, Dr. Benny. If something comes up, then our patients would love to know about this. And did you have a study on already a diabetic group on metformin with TNBC? Does their metastasis level, like progression, is lower compared to the TNBC group with no metformin intake?
The whole point of metformin as an anti-cancer drug came just like that, some correlation studies, then they found that there are some advantages, but again the detailed study is not conducted because those patients had a lot of other so I think those type of larger studies are needed now because because there may be a lot of other factors may be affected, like, because
Right.
metformin maybe they may be taking other things also. But there are some advantages already, which is known for
Okay.
metformin.
For
Yeah.
metformin.
Yeah.
That's great to know,
Yeah.
actually. Thank you. Thank you,
Yeah.
Dr.
Yeah.
Benny.
All right, Bill, you have a question?
Yeah, actually two. One has to do with as you introduced your presentation, you talked about the fact that you were using patients largely the African American community because the occurrence of triple negative was higher there. And I was wondering if, in fact, the occurrence of that triple negative cancer is more prevalent in African population or other smaller groups, for instance, Ashkenazi Jews or Scandinavian populations, where we've got a smaller population that's more self-contained.
Yeah, so the reason is I was talking about the racial disparity in triple negative is the studies have shown that even though triple negative maybe the prevalence may be more in Caucasians, the aggressiveness of triple negativeness, the tumors are more in African Americans. That is why, in NIH is now giving a lot of importance for a racial disparity in this some of these cancer types, like, because they are getting more aggressive type of cancer. They get young patients. They are more young patients. They get an African American patients, but I'm not sure whether we have, and also in Hispanic side we have we know that they are also having aggressive tumors, but there's no much studies focused in that direction because most of the clinical trial patients and most of the clinical trial samples are from Caucasian side. So now we're giving a little bit more importance to understand on the other side also. For example, I can say that in breast cancer cell lines, not even a single cell line I could find which is from Hispanic population. Because probably these all are derived earlier and who participate in the studies, all that makes a difference. So there is a lot more to do on, because we are also trying to see. One of our projects in the lab is to find out the racial disparity in cancer. Like, I'm not saying one group is less affected, the other group is more affected. Maybe each group has a different pathway which is responsible for that. So maybe a more, targeted therapy. Like, for example, in our case, for example, we found that the Caucasian triple negative, they have a high dependency for fatty acids metabolism, but not that much in the African American population. And the Caucasians or the European Americans are more fatty acid metabolism is depending on. So we need to do that type of a comparative study because there's a lot of genetic factors are controlling these tumors. And your question on other populations, I think there's a lot more to do because all these many of these studies are with a limited number of cases. So we need to increase the cases to understand the clear pathway.
Right, and part of what drove my question is the fact that we're finding out just in the last couple of years that although male breast cancer has traditionally been treated the same way that female breast cancer is treated, that there are some genomic and even subgenomic differences. And there are certainly differences in the way men respond to different protocols and treatments as opposed to the way women do. And so, what was originally thought of as being just a generic breast cancer is now really being found to be with differences between the genders. And I'm wondering whether if that in fact is the case between ethnic backgrounds as well.
Sure. Yeah, I was talking to Peggy many times this issue because there is very limited resources available about male breast cancer patients. Like, because if I have to study, there's no cell lines available, there's no tumors available to study, right? Because all these studies need to be done from a genomic level or genetic level, means we require material, and 2 or 3 will not work. Like, we need to do a larger study, or maybe at least 100. We need to study what makes them different. Why some men get breast cancer? Like, why not someone not getting breast cancer? So these type of studies need to be done. But again, you need to, thing is we need to increase our voice to, get more attention in that direction, because because always, when you consider breast cancer, we consider mostly it's a female disease. When you consider prostate cancer, we consider it's a male disease. That's the way it is categorized. So at least that's the way the impression come in the first. So probably more and more. And the first thing is, especially when you talk about a genetic difference, what you mentioned, to understand that we need samples, tissues, or maybe cell lines to understand, the basic character difference, why they are different, why someone is getting this problem. I think the groups like this should involve more in donating tissues, or maybe more involved in generating cell lines, or maybe some pilot funding to work on it, to understand that, because until and unless we don't understand the basic mechanism, we will continue with the same treatment.
Thank you
Because-
very much. Dr.
Because the
Benny.
companies may not be interested for a small population, right? Because of course they are interested, but the thing is they don't want to do a big study on that, right? So
Right.
if you understand the mechanism, maybe they develop a drug.
Right. Thank you very much.
Thank
for
you
your
so
comments.
much. Appreciate it.